Very nice. All right, good. So as Karen already informed you, I'm I'm a scientist working at the Netherlands Institute of Ecology. I dwelled around a little bit. I'm trained as a circadian biologist, and that's how I enrolled in a big project where we study the impact of artificial light at night. And how that works is that when I did work on circadian biology, I studied how the outdoor light dark cycle synchronizes the activity of nocturnal mammals. And as such, I got very interested in artificial night lighting. And some 10 years ago, we got a big grant and we set out with a big project on this topic. More about this later. But I have to inform you that as a secondary school pupil, I already was interested in bats really a lot. So I dwelled around by myself alone in the forest. With a bat detector, finding out that I couldn't tell all species apart with it. And therefore, I pieced together a tiny device where I could photograph flying bats, and it took me forever to get this going well. I sat in this old ruins. It's actually a castle destroyed in the Second World War. The basement is still intact, and I sat there trying to make photographs, and eventually it worked out really well. Anyway, so that's for my background on bats, because I just like these animals really a lot. Light at night. Light, as you may know. I'm not sure about Ireland, but here, if I walk in my backyard and look up in the sky, it's completely impossible to see the Milky Way, just see a number of stars, and that's it. So, and that holds pretty much for all of the Netherlands. And just to give you an impression, like most of the population about 99% of the population in Europe and the US lives in areas where where artificial light always exceeds the intensity of natural light, and it includes full moon. So just to give you an impression about how much light we have at night, is actually increasing by a good 2% per year. Now, artificial light has many different forms, but has two main forms. And well, one of these, if you can see my mouse here, is the simple attraction of animals, here insects, up here as well, these are birds. This is just like you can see in summertime, especially near aquatic structures. These are birds trapped in the 9/11 memorial lights in the southern tip of Manhattan. It's exactly the same mechanism. Animals are disoriented and are attracted and exhaust themselves, and in many cases actually die. A much more subtle way of light pollution is sky glow. This is a photograph I took in the periphery of one of the big cities in the Netherlands, and then what you can actually see is the light that's being reflected by the overcast. And as you can figure, you don't need a flashlight to find your way in the forest right here, which actually has a much more subtle effect. On many species, these effects are present, but they can also be more in the sort of a spatial way. And then you should think of the following example, which holds true for bats a lot, of course, but also for amphibians, for mustelid species, and also for insects, as we expect actually. And that is when a flyway or a route of animals is actually crossed by an illuminated road, which actually just simply blocks those animals from flying from one area to another. And of course, for bats, bats are roosting together in one roost, so often in decent numbers. Of course, it's impossible to go forage in one place, so they have to disperse, and such a barrier actually can deprive them from larger areas. So in this way, it's an amplification of the effect. Now, oh yeah, and a very nice, this has been shown very nicely, for example, by Emma Stone already some years ago. She illuminated hedgerows where she looked at different species of bat horseshoe bats, but also bats of Myotis, the Myotis genus, like this Nutterus bats. And as you can see here in this graph, light has a very strong impact on these bats commuting there. Here you see a measure of the number of bat passes per night. These are consecutive nights. In control night, you have a decent number of passing bats. If you present them noise, there's seems to be a slight but insignificant reduction. However, if you present them low, medium, or high lighting conditions, then these bats are shying away. So it is really, really important to be very careful with light, so to reduce light. And that can be done in several ways, very simple ways, often just like shielding light such that it doesn't spill over to areas where you don't want to have any light. So you keep the surrounding areas very dark. You can also lower the intensity, and quite often it's so that people can do very well with like half the intensity of the light intensity that was actually planned. You can also say, well, we only illuminate when there's actually traffic, or illuminate only in the evening and not later at night. And these are all good. But when we set out with our big project, we were mostly interested in another option of reducing the impact of light on bats, and that is by adjusting the light spectrum, the color composition of light. And this is simply because, like in the old days, well, talking like 15 years ago, we were sort of constrained to the traditional lighting light sources like high and low pressure sodium and halogen light and methyl halide, which have their own color composition. And of course you can tweak the color composition there, but you can only do so by placing filters, and actually that just means throwing away energy. Like municipalities and lighting engineers are not very inclined to actually use filters for changing the color composition. However, with the new LED lighting, it's much easier to change the color composition, and also to do so without throwing away energy. So you can just simply use white light, which has a balanced color composition using LEDs, but you also can use a green light with increased blue and reduced red component. And you can also use red light with an increased red and reduced blue component. And if you pile up all the information we have on the responses of animals, if you really strongly generalize, it's either end of the spectrum that may cause trouble for most of the species. I'll get back to this a little bit later. Already right now. So talking about nocturnal mammals, it's interesting to know that nocturnal mammals are not so well able to see red light. And I illustrate this here. So I have 4 graphs in a row here. On the x-axis and the horizontal axis, you see just the colors of the rainbow. So you can, this point, I plotted it here, but these colors are here too and here too. That's just the same as wavelength. And here, these graphs depict the response of the different photopigments of the human eye. So we have 3 types of cones, and we also have rods. And as you can see, we have a red cone, a green cone, and a blue cone. However, mice, and bats as well, and many nocturnal mammals, just simply do not have a red cone, so they're not as well able to see red light. Whereas they're very well able to see actually blue light, and in many cases even the shorter wavelengths, so that get close to UV light. And if you take the integral signal of the eye, which is not photopigments anymore, but is the electrical response that an eye produces when you put light on it, then this would create this graphs for different species of bat. And I made one of these graphs. I made blue because it's the Aptesicus fuscus, which is like a family of our Aptesicus cerotinus, a species that I expect also to be present in Ireland. Not entirely sure, but it's very common in the Netherlands at least. And if you look at the eye response, you can clearly see that it's very strong in the blue part and not so strong in the red part. And there's also another graph I'd like to show you because it's an important one if it comes to bat foraging behavior, and that's the action spectrum for the attraction of insects. Same on the horizontal axis, but here you just see the relative attractiveness of light, of this light color on insects. And as you can see very clearly, it has like the blue part of the spectrum has a very strong attraction it's very attracting insects very strongly, whereas the red part isn't at all. And if you wonder why the nocturnal species actually respond so strongly to blue light, it's simply because if you want to see something at night, you better have an eye that is sensitive for blue light. And you can very easily see this in the evening. If you look up at the sky, the dome of the sky is simply blue. So the light color composition is very bluish, and that all has to do with the light that is that goes at the side of the Earth, and it's being reflected by dust in the atmosphere and in space, and it's being reflected back to Earth. And typically the blue light is reflected that way. So it means that these animals, so it's not said that bats really can't see red light, but they can see it much less so than blue light. And it's also very important to keep in mind that the response of predictor for the behavioral response. For bats, there's not really much known about this, but for example, for turtles that hatch from eggs in like buried in tiny pits in beaches, and they have to make it back to the ocean. They are distracted by light really strongly. Their eye is very sensitive to the redder, the yellow, the green-yellow part of the spectrum, whereas the behavioral response is very strongly to the violet and blue part of the spectrum. So even though the eye is not all too sensitive for the violet-blue part of the spectrum, it relays this information anyhow, not very strongly, but the response is very strong. Anyhow, so talking about bats, it's very important to know why bats react to light so strongly, and it all has to do with predation. Of course, I mean, but like bats have a typical niche. They forage at night. You have insects at night, but relatively few predators, and it's widely believed that like the fear of light by bats just has to do with the fear of predators. It's very difficult to test though, but there's examples. For example, these bats that have to make their way out of caves where they are with millions of deer. There's a trade-off, of course. If you wait too long, you don't have so much time to forage, but if you're too early, then you're still in the light and you can be caught by bat falcons, for example. Very nice examples closer by are from Jens Riedel and Garrett Jones already some years ago, where they looked at the emergence time of bats in the evening and their flight speed. So here you have the time of emergence in minutes after sunset, and here you have the wing load, which is a sort of a measure for how fast bats fly, how much. Well, it's actually how much air the bats actually push downwards, but it's also a measure. It's a proxy for flight speed, and as you can see, the fast flying bat species, such as Nyctelis noctula and Nyctelis lazuli, for example, they emerge very early because they are very fast flyers and are difficult to catch for predators, whereas the Myotis species and Plecotis species, they wait very long before they actually emerge. Another very nice example is for pipistrelles. It's the same kind of graph. Here you see the emergence time in minutes after sunset. And here you see the time in days before and after giving birth. So these female pipistrelles get heavier and heavier, and hence can fly less fast than normal, and also are less agile. So they emerge actually later. And then, of course, when they give birth, they lose a lot of weight. They can fly much easier, and they start to emerge back earlier again. So that's a really clear indication that they are that they avoid predators, and that this avoidance is dependent on how well they're actually able to escape those predators. Well, to give you some examples of bats here, and also how they respond to light. So here we have the slow-flying bat species that like to forage in the vegetation. These were the ones to emerge late. They're very shy of light, very shy of predators, obviously. But there's these pipistrelles, for example, that fly much faster, are much agile, and also dare to forage in the open. And then, of course, you have here ceratines and the Nicholas species. They fly much faster, they're much stronger, also dare to fly in the open. And if it comes to artificial light at night, then pipistrelles are more than happy. Even though they don't like that light very much, they're more than happy to forage on these accumulated insects around these light sources. Now, in terms of biodiversity, it's important to realize what happens if the amount of light at night actually increases, because what we then do is actually facilitate already abundant species, at least here in the Netherlands, and we, like hamper, we deprive the slow-flying species, which are much less abundant actually here and in most of Europe, from their foraging habitat. Now, what we did is we set up a big experiment, as I already informed you a little bit, where we tested the impact of light of different color in pristine nature. So we experimentally did this. We just put light posts in forest edges and tested the response of many different species. Now, there was a bit of an effort because, well, the Netherlands is a sort of developed country, but if it comes to electricity in nature areas, there's still really a lot to do, and so had we. If you want to put light posts, of course, you need electricity, and that, yeah, well, in nature areas that need to come from some distance sometimes. But eventually, we ended up with, in 2012 already, with a very nice setup where we had a combination of rows with light posts, as I explained earlier on, white light, green light, and red light at 8 locations in the Netherlands. So at 8 replicated sites, we had combinations of each of those. So in total, we had 32 rows with light posts of different colors. And we measured the response of bats for 6 consecutive years. And of course, we did so with automatic bat detectors. Yeah, so okay. But this is what it looks like actually. So it's quite artistic, I would say. So if you measure the response of bats there for a prolonged period of time, then the response of bats looks like this. So here you have a measure of the total number of passes, bat passes per transect, and here you have the different light treatments. And this graph goes for the slow-flying bat species. In the dark, you have relatively much activity for these species. In the red light, you have equally much activity. Equally much activity compared to the dark, but in the white and green light, the activity is significantly reduced. For pipistrelles, the agile species that are not so afraid of light, even though they don't like it, there's relatively little activity in the dark. Well, bear in mind the axis here, because these are much less abundant. You can tell from the numbers here, but relatively little activity in the dark, equally little activity in the red light, significantly more in the green and in the white light. Then we have the ceratines and the noctules combined. it sort of shows the same pattern as the pipistrelles, but you can tell from the variation we got there, we can't really say something there because the data aren't significant. Talking about the pipistrelles, this is all driven by the insects. As I put this graph I showed you earlier on here. I put this below here. So the red light doesn't attract many insects, as you can tell from this graph here, whereas the green and the white light is all lights combined, of course, attract many more insects. And actually, they fully explain statistically significant the increase of pipistrelles in the white and the green light. The response of the slow-flying species, on the contrary, is mostly due to the fact, as I explained earlier on, that those species are less sensitive for red light. Now, this is like foraging habitat, and it may be the response of bats, depending on their behavior, may be completely different. And in the Netherlands, you have like aquatic structures are very, very important flyways for different bats, and we were very curious on how different bats flying using these flyways actually would respond to these different light colors. And actually, so not far away from our other experimental sites, we had this excellent situation where we have Daubenton's bats flying from their colony to their foraging areas using two identical culverts. And each night, like about 40 bats were flying this way, and in the morning they were flying, of course, the opposite way. So we equipped those two kinds of light, and it looks actually like this. All light types were in there, so green light, white light, and red light. Same here. And we just could switch these light colors by mobile phone, and so we could easily implement the protocol. We fitted those tunnels with automatic bat detectors and actually measured the bat activity right in there. And we did this for 45 consecutive nights, and such that we never had the same light condition in 2 consecutive nights in the same culvert, and always had a combination of 2 light treatments, so bats really had to make a choice where to go, left or right. And of course, they were equally balanced to counteract any natural preference for these bats for one of the 2 culverts. And to our big surprise, these bats didn't respond to light at all. So here again, you see a number of passing bats, and here you see the different light treatments. Well, first of all, you can see there's no change compared to dark, whether there's light or not, but there's also no difference between the white, green, and red treatment. This really surprised us a little bit. To some extent, because you have to know that some other people actually already did some work on pond bats, where they unfortunately only at 2 sites. Well, of course, our Daubenton's bat study was at 1 site, which effectively means it's an NLS1 study. This was done at 2 sites, comparable to Emma Stone's experimental setup. They measured passing pond bats, Meodus during one dark night, followed by an illuminated night, and then a dark night again. And as you can see, so these are 2 different locations. So the dark rods are one location, and the white rods are one location. And as you can see, there's no effect at all. In fact, in one location, there were more passing bats during the illuminated night compared to a previous dark night. It doesn't tell a lot, but at least the effect there wasn't so strong that all the bats turned around or just didn't pass. It contrasts with, of course, with Emma Stone's study that I showed earlier on, with the bats passing along a hedgerow, and it also contrasts with a newer study by Manisha Bhartwai, who looked at bats passing under underpasses and culverts, which she also experimentally illuminated, and she saw a very strong effect there as well. So there's definitely more work to do there, and I'll get back to this. We're going to do that. It's, however, interesting to look at in a study by Matt Zeal some years ago, not so many years ago. It's comparable to Emma Stone's work, where he illuminated the same hedgerows where she did her work earlier on with different colors of light, like we did, slightly different spectra. And that shows about the same picture as we got from the experimental light posts we put in the forest edge. So relatively much activity in the dark, comparably much activity in the red. So it's like you can see the statistical significance here. Significantly less activity in the white. And then you see some reduction in the green and orange. And talking about the orange, you see here red light and orange light. Of course, it's on the red side of the spectrum. I'd like to say a little bit more about the amber lighting you may have heard of. So amber lighting, and at least in the Netherlands, was branded as the bat lamp, which I get a lot of questions about, and which still is sort of a difficult story. So in 2011, the Dutch Mammal Society did an experiment with amber lighting. It's a light that has a narrow band frequency of about 591 nanometers. I have to say the red light we use has a peak at 610 nanometers, which is more red than the amber lighting. And what they expected was if they would like put lights at the side of a kennel. So this is a kennel here, right here. There's pond bats flying from their colony to their foraging grounds. And if you put lights there, you would actually push those bats to the dark side of the kennel. Whereas if like with amber lighting, this deterrent effect of light on bats would not be as extreme. Unfortunately, they did not observe this effect. So I mean, this is a hypothetical picture. In fact, the effect of amber lighting was exactly the same. However, after this experiment, the commercial like companies started selling those lights and actually sold them as bat-friendly lamps. They may actually be bat-friendly, but not for sure. And then it's also interesting to look at a study by Straker et al. It showed up last year. It's from the lab of Christian Voigt in Berlin. And one of the things they did there was like test bats in flight chambers. So it's a very simple approach. You have two adjacent bat chambers where the bat has a choice where to fly. And you can switch the light conditions between those two chambers, and you can test different lights against each other, just like we did in the culverts. Now, here's two species of bat where you have here your control. Like if the dot is right on this middle line here, then the bats don't have any preference, and so this is just control, dark against dark. And here you have amber against red, red against white, and white against amber. And as you can see, the red light here is preferred much more than the white light, whereas with amber, this effect is not statistically significant. And again, since again here was Myotis capucini. So it does show that maybe the amber lighting is not red enough for these bats. So it's maybe still too much within the sensitivity of the eyes of bats. And there's many on. I can tell forever. I won't be talking so long. But there's many other interesting things, and just name just a few of them. What I find very interesting is the response of pipistrelles to light, for example. So James Hale did some experiments there where he looked at the crossing distance for bats between vegetation structures. So they had to cross an open space. And he looked at whether bats turned around or whether they successfully crossed the gap in this vegetation. so here, the blue ones here are the successful crosses, and the red one are the attempts of bats, but they didn't successfully cross the gap. And as you can see, many more bats successfully cross a gap if it's short, but also many more bats cross a gap if the light intensity is lower. In fact, they are able to cross much wider gaps if there's less light, less ambient light. Super interesting, because these pipistrelles are happy to forage on the accumulated insects, but yet they don't like the presence of light when behaving, when expressing other behaviors. Then there's some information, but it's rather anecdotal, I would say, on the response of migratory bats to light. But however, these studies are still quite anecdotal and really need to be replicated, but super interesting. All right, and then also to inform you a little bit on the response of bats when flying out of colonies. It's well known that bats in colonies are also don't like to emerge when there's light, as I showed earlier on. In the old days, which is super interesting. Anyway, so in the old days, they actually used lights to get rid of bat colonies. It's really a long time ago. So here you see the development of a colony across the season. While it's increasing in numbers, as you can see here, it's like approximately the end, halfway June until the end of July. That holds, like that's on the horizontal axis, and also here. If you put light there, the colony, you see a decrease here, a decrease here, and a decrease there, whereas the dark control actually goes up. If it comes to different light colors, there's very limited information. There's a nice study by Downs et al. Where he counted the number of emerging bats in different lighting conditions. It's two roosts. The two different bars are two roots where different colors of light are actually presented, and as you can see, there's really much variation there. And yeah, you can tell very clearly from this graph that we really need more information. We need to test many more roosts and see how bats respond to different light colors. Well, you may derive from what I just said about the different light colors that the red part of the spectrum is probably much better for bats than just white light, or worse, the green or blue. However, when using this in real life and advising on municipalities on where to use different light colors, it's very important to realize that these red lights are not as effective everywhere. For example, at least in the Netherlands, if you are in built-up areas with a lot of ambient lighting, these slow-flying bats like Plecotis and Myotis are not there anyway. So it's like placing red light there is not all too useful. It's also important because it has proved in the Netherlands that not everybody likes this light. If you inform people, it works much better, but eventually people really prefer white light. Whereas in the countryside, where you have those species and where lights actually may form a barrier, the placement of red light is much more effective. But I still like to warn you about the next example, because if we know that slow-flying bat species dare to fly close to a red light, it may actually form an ecological trap, because they don't experience it as a lot of light, because they can't really perceive it well, but they may be seen by predators relatively easily and actually like expose themselves to these predators. So that's something that we think is very important to think about. Even though there are not so many records of like, for example, owls taking bats in midair. All right, I'm nearly at the end of my talk. I'll just tell you a little bit of what we are currently doing. One of the things is that we like to learn more about the response of bats to light intensity, because it's a question we get all the time from municipalities and light installers. So what light levels should I keep below, not to destroy habitat for bats? And in order to know this, you really have to know how bats respond to light. And one of the things we use is our microphone arrays, where you use several different microphones, and actually you make use of the echolocation of bats, and you look at time differences between the arrival of the echolocation pulses at the different microphones, and you can actually calculate where the bat actually was when it emitted that pulse. And as many species, as you know, emit anywhere between 5 and over 10 pulses per second, you get a very accurate a track of actually flying bats around light sources. And just by looking at the light intensities, because you know the light intensity because we measured it, you can tell, for example, threshold levels to which bats actually tolerate light. But one of the things we saw is, like, if you follow bats around light sources, as closer they get, the more they get into the vegetation. And that's very interesting, because they actually seek refuge in vegetation. That's what we think they do. So we did a first experiment with Kevin Barret, and actually it really looks like it really showed that bats actually fly into the vegetation. And we're currently looking at this much more closely now, together with Claire Hermans and Jens Koblitz, using these microphone arrays and actually making LiDAR scans of the vegetation. It's a method to actually very narrow and very accurately pinpoint all objects in a space, and you can actually scan a whole forest in that respect. It's interesting because, as you may know, that many bats aren't able to catch insects all too close to the vegetation, for the simple reason that the echoes of the vegetation will exceed the echoes they get back from the insects. All right. We're doing the same also with passing pond bats. It's very much like the studies already showed you, but now not only at 2 sites, but at no less than 20 different sites with different light colors, and where we again use these microphone arrays to exactly see the response of these bats to light. We can actually see whether they turn around or find darker areas, for example, right here to fly through, so we can really plot the response of those bats to different light levels as well. All right, so there I'm at the end of my talk. To summarize a little bit, well, of course, it's very important to reduce light. It can be an option to use red light, and then specifically in natural rural areas. If people really want light, then it's best to, of course, use low intensities, but then also use warm white light. That's usually expressed in degrees Kelvin, for which you should use less than 2700 degrees Kelvin. It simply means, like, the lower the Kelvin number, the less blue is present in the light, and the less blue, that is actually better because it's perceived as less intense by that. Green light, at least in the Netherlands here, it's installed at some areas. I didn't inform you much about it, but green light does not disorient migrating birds at night at sea, but it's only that situation. So if you're onshore, if you're dealing with terrestrial habitat, green light is never a good option. But bear in mind, the best solution is location specific. And with that, I'd like to end my talk, and thanks for listening. And I'm happy to take questions.
What is the public perception to red light zones?
Yeah, so that's an interesting one. So we have, if I can share my screen for. So I took one example. Hang on. I took oh dear. Do it again. There we are. So I had one example where we have a red light in. So there's like a few examples. Sorry. So there's a few examples of where red lights were installed in like inhabited area. And at several places, actually, people complained about the red light because they didn't like it. And quite frankly, I don't like it myself as much either. However, the acceptance rate of this red light is dependent on how well you inform people. So, for example, here you see an aerial photograph of one of the wetland areas in the Netherlands, where there is a pond bat migration route, and I think it goes right here. So a number of pond bats fly here, and a number of pond bats fly here as well. They disperse a little bit. So what they did is they installed the red lighting, I think in this area here, this street here, in order to mitigate the impact on pond bats. Even though we're not sure of the, we didn't advise there, but well, I mean, supposedly it works for pond bats. We still have to find out though. But yeah, so here that's a clear example, and people were informed, like, hang on, these are pond bats, and the Netherlands has a special responsibility if it goes for pond bats. So like the core of the population of pond bats in Europe is in the Netherlands. So we really have to be careful not to lose numbers of them. And there they accepted the lighting. So yeah, it's a matter of informing people, and then it works much better. But it's also like really making a good plan. And yeah, realizing that, like in most built-up areas, it's not really necessary to install red lighting at all. But in rural areas, it may be a much better option.
Yeah, I was just wondering about the wavelengths. I know you gave 600 nanometers, was it, for the red light, Kamiel? Was there any variation in the available LED types, red LEDs? Or was the 600 nanometers the recommended?
Yeah, that's a good question. I hope I have. Yeah, so I have some of the wavelength here. So here, if you can see my the cursor of my mouse, yeah, these are the wavelengths we actually tested. This is the green lighting here, the white lighting here, and the red lighting we tested here. First of all, if you look at the spectrum, you see that the red light here contains a huge peak of red light. And I have to inform you that, like, even though we ourselves can see red light much better than nocturnal mammal species, we know we can see it as well as green light. I mean, like the optimum in our vision and human vision is green. It's 555 nanometers. It's right here. So in order to make streetlights that are equally intense, but are red light, then you have to crank out really a lot of red light. With amber, because it's closer to the middle here, the peak is lower here already. With white light, because it contains all these wavelengths in a sort of balanced form, there's not a single frequency here that has such a peak, because it doesn't need to, because with white light it's very easy to make a light that's equally intense. And then with green light, it's sort of comparable, because we're very sensitive to green light. So to get back to your question, the only thing we know is that, like, in the experimental sites where we put these light posts out there, we know we know very sure that these red lights just like do not affect the activity of the slow flying species. And as you can tell here, the peak in our red light is well above 600 nanometers. Whereas in the amber lighting, it's below 600 nanometers. So yeah, I already told you a little bit about it. The amber lighting may be a bit too much to the middle of the spectrum there. And that is as much as we know. It's a bit of the same story with Matt Zeil's work, where he studied the impact of red lighting and then also orange lighting. So the impact of red lighting on commuting bats was less than orange lighting. And as you can tell here, I put a tiny line here, and as you can see, the peaks here are more to the middle than is the peak here and the red lighting here. So it may be the same story as I explained right here. And this is pretty much as good as it gets. You know, as a scientist, you'd like to test every slice of the spectrum, every nanometer, and then all combinations. But you know, you can imagine how much work that would be. As for now, it looks like if you want to avoid the like affecting activity of slow flying bats, then you really have to be above 600 nanometers. Yeah.
Okay, thanks for that. I think it was Herman Limpens who many years ago had recommended orange lighting at 591 nanometers, but I dare say this research surpasses it.